Okay. So I would like to thank RSGDREAM organizer for giving me this wonderful opportunity to present our latest research work on phylogenetic dynamic regulatory module network to study evolutions of mammalian gene regulatory program. We know that cis-regulatory elements such as promoter and enhancer are abundant and drive the gene expression level. Transcription factor bound to promoter and enhancer are a backbone that constitute the gene regulatory networks. Changes in gene expression and its regulation significantly contribute to morphological diversity across species. Beyond sequences, comparative regulatory genomic studies collects and compare multiomics measurement from different regulatory signals. Comparative regulatory genomic studies allow provides the systematic study of the gene regulation across species to infer the evolutions of the complex trait. So right now, certain studies have been performed that incorporates the expression data and the phylogenetic relationship across species to infer the species-specific expression modules, regulatory networks, and the subnetworks. However, these studies lack the integration of multimodal omics data. On the other hand, certain studies have been performed that integrates the multiomics uh, signals from different transcriptome and epigenome assay to predict the gene regulatory networks in certain contexts like disease progression or the developmental phases in certain time points. However, computational tools that are phylogenetically aware and can measure such high-dimensional multiomics measurement across species to infer the evolutionary gene regulatory network remains scarce. Here, we are introducing phylogenetic dynamic regulatory module networks, P-DRMN, which is a novel multitask regression-based learning algorithm that predicts the dynamic gene module uh, regulatory networks by leveraging the bulk multiomics data and the phylogenetic relationship of the studied species. Before going to P-DRMN, now I'm going to tell you about what is the regulatory module network for one species. So in regulatory module network, we represent the modules as a set of genes, and these genes are represent a particular expression state for a species, and which we can interpret as a discrete expression level. Next, in the regulatory module network, the key component is the regulatory programming, which is defined by a collections of graph structure, that is G, means what are the regulator for gene expression, and a parameter that estimates how a regulator predict the expression state of a gene module. So as we are using the simple linear regression, so the parameter would be regression coefficient. The measured activity for a regulator is the signal strength of the transcription factor and the cis-regulatory element for the gene that present in a module. So this is how we can incorporate the multiomics chromatin data into our regression model. Further, we train the model to predict the expressions of the gene modules by its regulatory feature vector. So P-DRMN is a multi-species version of the regulatory module network that integrates the multiple signal from multiple species and models the gene expression at the level of similarly expressing gene module based upon the low to high expression of the input gene, and it predicts the dynamic gene regulatory module network for each studied species, where a gene membership in a module is based on the regression function of its upstream cis-regulatory features. Our collaborator, Professor Michael Wilson from Sickkids Hospitals, is working on the endothelial cell system. Endothelial cell system lines our blood vessels, involves in the vascular homeostasis, responds to injury and inflammation, and forms the blood vessel. So endothelial cell dysfunction is implicated in several diseases, including the cardiovascular diseases, atherosclerosis, and the irritable bowel disease. Wilson lab have already prepared the basal level aortic endothelial cell data from different multiomics methodology, including the RNA sequencing, ATAC sequencing, and chip sequencing of the 5 histone marks for the 5 mammalian species: human, rat, cow, pig, and dog. So we use this data for the P-DRMN modeling to infer the evolutions of gene regulatory program in the aortic endothelial cell of these 5 mammalian species. P-DRMN application identified certain gene modules. So here we can see that each box is representing a gene module. The blue color indicates the low expressing gene module, and the red color indicates the high expressing gene module. And the number represented here is the number of constituent genes that present in a gene module. And we found that in the aortic endothelial cell of these 5 studied mammalian species, having the conserved gene module pattern across species. Further, we identified the predictive power of the P-DRMN inferred gene expression module by calculating the correlation between the actual expression state and the P-DRMN predicted expression state. And we found that an average correlation of 0.39 to 0.53 was achieved. Interestingly, in certain modules like that module 2 of the rat, the correlation score reaches 0.76, which indicates that P-DRMN can predict the actual expression state of the gene modules very well in certain species. Next, we calculated the module conservation score using the Jaccard index of the gene module assignment for a module across species. Like for module 1, we calculated the human to rat, human to cow, human to pig, and so on. And we found that a conservation score of 0.19 to 0.65 was achieved. And among these, the high expression gene module like module 3, 4, and 5 exhibit a greater conservation across species, whereby the low expressing gene module like module 1 and module 2 having the significant divergence across species. Next, we try to identify the biological function exerted by this conserved gene module of the aortic endothelial cell. And we found that the low expressing gene module means that repressive gene modules are associated with the non-endothelial cell specific biological functionalities, whereby the highest expressing gene module, that is the module 5, is associated with the important aortic endothelial cell function, that is angiogenesis, means the new blood vessel development. And after that, we identified certain genes that changes the module assignment based on their expression state. For an example, like HOXB3 gene that having a low module assignment in all the studied species except human. In human, it changes the module assignment to a higher expression state. So HOXB3 is a classic example of the transitioning gene. The set of transitioning genes that follow the similar pattern, same pattern across phylogenetic species and the clade are known as the transitioning gene set. In the aortic endothelial cells, from these P-DRMN inferred modules, we identified a total of 103 transitioning gene set across species and clades that having a diverse phylogenetic trend among the species. And here now I am demonstrating two interesting transitioning gene set. So one is the human specific transitioning gene set and one is the cowpea clade specific transitioning gene set. In the human specific transitioning gene set, cluster set 55, in human, it changes the high module assignment compared to the other species. And in the cowpea clade specific transitioning gene set, that is cluster set 86, where 17 genes changes to high module expression module assignment compared to other species and clade. So further, we infer the putative regulator for this transitioning gene set such a way that if the regulator expression is correlated to the expressions of the at least 5 gene in the transitioning gene set with a Pearson correlation value of 0.6 and higher. And by this way, we identified that a human-specific transitioning gene set is predicted to be regulated by CTCF. And we all know that the CTCF is an important regulatory protein that involves in the regulating the chromatin architecture. And for the second important transitioning gene set, we identified that SOX2 is a putative regulator as well as the two histone marks, including H3K27Me3 and H3K4Me3. So a combination of transcription factors and the histone marks contribute to the changes in the expression across species. So taken together, P-DRMN provides the powerful framework to leverage the multiomics measurement across species to systematically examine the evolutions of gene regulatory programs and networks. That's all for today. I would like to thank Roy Lab and Wilson Lab member for their help in each and every possible way. And I am also thankful to Wisconsin Institute for Discovery and University of Wisconsin-Madison for their high throughput computational and research facilities. I am thankful to my postdoctoral funding agency, Department of Energy, and the James S. McDonnell Foundation for providing the necessary financial support. Thank you all. If you have any question, let me know.